Well, hey there. Today we're tackling DNA, life's blueprint, and how it changes over time, how it evolves. Right, it sounds complex, and it is, but we've got this analogy that really helps. The river of life analogy, exactly. Picture DNA as a dry riverbed, specific paths within it, the channels, those are your gene sequences. Okay, the A's, T's, C's, and G's lined up precisely. And gene expression, that's like the water actually flowing through those channels. And evolution, that's the riverbed itself changing over long periods, maybe erosion, maybe a landslide. The whole structure alternates, got it? So the aim here is to use this visual to get a solid grasp. Let's dive in. So the riverbed itself, the whole structure, that's the DNA molecule storing information, and the channels are the genes. What about the physical structure, the double helix stuff? Think of it like the material of the riverbed. You know, rocks, sand, clay, how it's packed together, that higher order structure. Effects, which channels are even accessible for water flow. Okay, it makes sense. So what gets the water flowing? What starts gene expression? That's the cellular machinery. Key players are things like RNA, polymerase, ribosomes, the cell's workers, and the water flow itself is the process. First transcription, then translation, leading to the actual functional products like proteins. That's the river actually flowing and doing something. So RNA polymers, think of it like a specialized drone. Finds the start of a gene channel, a starting marker. It binds there, unwinds the DNA a bit, widens the channel, if you will. And then it reads the DNA sequence and builds. It builds a messenger RNA, molecule, and mRNA copy. It's like creating a temporary map of that specific channel's path. And it just keeps going until it hits a stop sign, a terminator sequence. Then the mRNA map is released. In our cells, eukaryotes, there's often a bit more to it, but editing the map, kind of. The first draft, the pre-mRNA, often has sections that don't code for protein called introns. Think of them as debris or blockages in the channel. They clear them out. Exactly, those introns are spliced out, and the important coding bits, the exons, the clear paths, plus you add protective caps on the ends, sort of reinforcing the map's start and finish. Finished mRNA map, that's what leads to a protein. Now we're at translation. The mRNA map travels to a ribosome. Think of it as a translation hub or an assembly point. The ribosome reads the map in three-letter words called codons. Each codon specifies a particular amino acid building block. Little delivery molecules called tRNAs, like feeder streams, bring the correct amino acid for each codon. The ribosome links them together one by one, forming a chain of polypeptides. This chain then folds up into a specific three-dimensional shape, becoming the final functional protein. The river takes its final functional form. It's quite deterministic, isn't it? The water really has to follow the channel laid out by the code. Largely, yes, although one thing the analogy simplifies quite a bit is regulation. It's not just about the channels existing. There are gates and controls, like deciding when and how much water flows. Transcription factors and other molecules act like those gates, controlling access to the gene channels. The riverbed isn't fixed forever, it changes. How does that happen in the DNA? Small scale. First, corrosion or sand shifting slightly. Those are point mutations, tiny changes. Maybe one letter swapped for another, or one added or deleted. Just small wear and tear. But then you have the big stuff. Rock slides, floods, changing the course. Those are like larger mutations. Deleting whole sections, duplicating parts, flipping them around. Major rearrangement. Just change the whole flow path. And sometimes the changes aren't even in the main channel sequence itself. You mean epigenetics, like adding pebbles or plants near the channel? Exactly. Epigenetic marks like DNA methylation or changes to how DNA is packaged by histone proteins. They don't change the A's, T's, C's, and G's, but they affect the flow anyway. They can act like silt, building up to silencing the gene or loosening the soil to make it more accessible. They control access to the channels. Absolutely, that's natural selection. The landscape is the environment. If it changed to the riverbed, a mutation happens to make the river flow better in that particular landscape, helps the organisms survive or reproduce better. Then that version of the riverbed is more likely to persist, to be copied in this next generation. The beneficial change becomes more common, so the landscape filters the changes. It doesn't cause them, but it favors some over others. Variation happens randomly, like the riverbed changing by chance. Selection is the environmental pressure favoring the fitter forms. But it's not always about being better, is it? Sometimes things just happen by chance, especially in small populations. Right, that's genetic drift. Imagine a random pebble falling and slightly diverting a tiny stream. In a huge river, it wouldn't matter much. But in a small stream, it could change the whole course. Exactly. In small populations, random chance, just sampling error from one generation to the next, can cause gene versions, alleles, to become more or less common or even disappear entirely, regardless of whether they're helpful or not. Like the bottleneck effect?
Yeah, like if a disaster drastically reduces the population size.
Do the survivors are random?
Maybe not the fittest.
And their genes define the new population's makeup.
Or the founder effect,
Where a small group starts a new population.
Right.
They only carry a subset of the original genetic diversity.
So the new river system starts off different, just by chance.
So luck plays a big role, especially for small groups.
a surprisingly big role.
Okay, so we have changes.
How do we get entirely new river systems, new species? Think about a river getting physically split by, say, a mountain range rising. Geographic isolation, that's allopatric speciation. The two river systems are now separate. They evolve independently, accumulating different changes through mutation, drift, selection. Well, till they're so different that even if the barrier disappeared, they couldn't flow together anymore. They can't interbreed. They become reproductively isolated, separate species. Can it happen without a physical barrier? Sometimes, yes. Sympatric speciation. It's trickier to visualize with the river, maybe like a sudden drastic change in the water chemistry within one part of the river, making it incompatible with the rest, like polyploidy plants. That's a classic example. A sudden doubling of chromosomes can instantly create reproductive isolation within the same location. It's an internal change blocking the flow between the new type. This river analogy really does map surprisingly well onto these huge biological ideas. It helps structure the thinking, doesn't it? DNA is the riverbed blueprint. Expression as the water flow, mutations as changes to the bed, selection is the landscape's influence, drift as chance events, speciation as diverging streams. It gives you hooks to hang the concepts on. But like you said, it's a simplification. Oh, absolutely. The molecular reality, the regulatory networks, they're incredibly intricate. DNA isn't static like rock, it's dynamic. The analogy breaks down if you push it too far. Still a really useful tool. So a final thought to leave folks with. Well, we talked about the external landscape shaping the riverbed through selection, but what if you consider the landscape inside our own bodies, like the cellular environment, our immune system? Could that internal environment act as a selective force, shaping our own genetic material, maybe even within different cell lineages in our body over our lifetime? It's a complex idea, kind of hindered by the analogy, but takes it a step further. Something to mull over, definitely food for thought. Thanks, this has been a fascinating deep dive.